Hello and welcome to Reviewing with Mrs. Wages. This video is meant to review objective number 2 of the DNA unit. Objective 2 asks you to explain DNA replication. So let's begin by explaining what replication is. What is DNA replication? DNA replication is the process by which DNA copies itself. One molecule of DNA is used as the template to build two identical strands of DNA. So where does replication happen in the cell? Well, DNA is locked away in the nucleus of eukaryotic cells. It's stored here to keep it protected. Thus, the copying process, or DNA replication, must occur in the nucleus of the cell. And when exactly does replication occur? Think back to the entire cell cycle. It's broken down into three major steps: interphase, mitosis, and cytokinesis. Interphase is then also broken down into three phases: G1, S, and G2, all of which are preparing for cell division. The process of DNA replication occurs during the S phase of interphase. Remember, the S phase stands for synthesis of new DNA. And why does a cell bother to replicate its DNA anyway? A cell replicates its DNA to prepare for cell division. Each daughter cell needs its own copy of DNA to function properly. All right, what does a cell need in order to copy its DNA? Well, it needs three things. To replicate DNA, the nucleus of the cell must have each of these three items: the original strand to be copied. This will serve as the template for the new strands. It will also need the enzyme DNA polymerase, which orchestrates the whole process of replication. And finally, it needs free-floating DNA nucleotides of every variety, A's, G's, T's, and C's, in order to build the new sides of each of the DNA molecules. Okay, so how does the whole process work? Well, replication essentially happens in three steps. Number one, DNA replication begins when DNA polymerase unzips the DNA by breaking the hydrogen bonds holding the chains of nucleotides together. This exposes the places where hydrogen bonds can be formed with new nucleotides. Number two, DNA polymerase then grabs free-floating nucleotides that are hanging out in the nucleus and binds them to the unzipped DNA on both sides. It makes sure to attach these nucleotides in the proper order based on DNA's code to form accurate and complementary base pairs. The third and final step occurs when DNA polymerase proofreads both new strands of DNA to make sure that there's no mistakes, which would be called mutations. When this process is complete, the chromosome containing the DNA will be replicated and take on that familiar X shape as seen at the beginning of mitosis. And now you should be able to describe the process of DNA replication. That's all for now, and as always, happy studying!